Hello, y'all. This is Pam. So for this week, I chose to explain what a test cross is and to give an example of how it would be used. I always like to start with definitions. It seems to help me a lot. Test cross is a cross between a dominant expressing individual with an unknown genotype and a homozygous recessive individual. The offspring phenotypes indicate whether the unknown parent is heterozygous for the dominant trait. Homozygous is having two identical alleles for a given gene on the homologous chromosome. Heterozygous is having two different alleles for a given gene on the homologous chromosome. Of course, an allele is a gene variation that arises by mutation and exists at the same relative locations on homologous chromosomes. Homologous chromosomes are chromosomes that share the same structural features, the same genes at the same loci positions. While the genes are the same, alleles may be different. Of course, we're talking about a test cross, and the test cross was devised by Gregor Mendel, and it's a technique that's still used today by plant and animal breeders. So a test cross can be used to determine the organism's genotype. So how that's done is the test is placed on a Punnett square. And what we're doing is we're using, for this example, a capital Y and two lowercase y's. The capital Y stands for the gametes from a parent of unknown genotype, and we're crossing it with gametes from recessive parents. And on this test, a test cross resulting in all dominant offsprings will indicate that a parent is homozygous dominant. Gametes from parents of unknown genotype cross with gametes from recessive parents. The unknown is capital Y. The lowercase is the questionable. And that test cross results in a 1.1 ratio of yellow to green. To orange. I didn't have green. Offsprings indicate that the parent is heterozygous. And this is my drawing again. All right, thanks a lot. Hope that helped.